Hey Year 11, it's Mimi
And so,
and this is our video.
on replication, transcription, and translation.
We hope you guys learned something with our video.
Replication, transcription, and translation. Introducing DNA. DNA is the genetic material of all living organisms. It is made of relatively simple subunits. These are called nucleotides. DNA stands for deoxyribonucleic acid. Nucleotides are composed of a phosphate, a deoxyribose sugar, and a base, like shown in the diagram. Two DNA nucleotides can be linked by a covalent bond between the sugar of one nucleotide and the phosphate group of the other.
The nitrogen bases are adenine, cytosine, thymine, and guanine. DNA molecules consist of two strands of nucleotides which are then wounded together to form a double helix. These are formed between the bases of two strands.
However, it is formed by complementary base pairing because adenine only forms hydrogen bonds with thymine, and cytosine only forms hydrogen bonds with guanine.
So that was just an introduction for DNA.
Interesting, right?
Well, now you're going to learn about replication. DNA replication.
DNA replication is semi-conservative. DNA replication copies DNA to produce new molecules with the same base sequence.
It is semi-conservative because each new molecule formed by replication uses one new strand and one old strand, which is conserved from the parent DNA molecule as shown in the diagram.
The unwinding of the double helix and separation of the strands by helicase, followed by formation of the new complementary strands of DNA polymerase, happens during DNA replication.
In order to replicate, the two nucleotide strands of a DNA molecule separate at their base pairs. Each strand then builds its opposite strand by base pairing with free nucleotides. For example, if one nucleotide on a strand has thymine as a base, the free nucleotide bonded to it would be adenine. Each new replicated DNA molecule has one nucleotide strand from the original DNA molecule and one nucleotide strand that has been newly synthesized from free nucleotides in the cell.
Now we have a bonus for you guys. Differences between DNA and RNA.
That you're going to have to know that for your exam.
DNA versus RNA structures. 1. The number of strands. DNA has two strands, forming a double helix. RNA has one strand. 2. The type of sugar. DNA has a deoxyribose. RNA has a ribose. 3. The nucleotides. DNA has A, C, G, and T. RNA has A, C, G, U.
Saw the differences?
Remember them.
Now we're going to have C transcription.
Have fun.
DNA transcription. Transcription is the copying of the base sequence of a gene by making an RNA molecule.
Transcription of DNA. The process of transcription begins as enzymes unzip the molecule of DNA, just as they do during DNA replication. As the DNA molecule unzips, free RNA nucleotides pair with complementary DNA nucleotides on one of the DNA strands. Thus, if a sequence of codons on the DNA strand were AGC, TAA, CCG, the sequence of codons on the RNA strand would be UCG, AUU, GGC. When the process of base pairing is completed, the mRNA molecule breaks away as the DNA strands rejoin. The mRNA leaves the nucleus and enters the cytoplasm.
The genetic code is a triplet code. 3 bases code for one amino acid. A group of 3 bases is called a codon.
And now, guess what? The most interesting part.
Translation!
Translation. Translation is the production of proteins by decoding messenger RNA produced in transcription.
Translation is the process by which the information contained in messenger RNA is used to direct the synthesis of a polypeptide. This information is carried in the sequence of bases on the messenger RNA molecule. The polypeptide can be assembled once RNA binds to a ribosome. Ribosomes consist of large and small subunits. The large subunit has 2 binding sites for transfer RNA, the P and A sites. Transfer RNA carries an amino acid, which will be incorporated into the polypeptide. Its anticodon is a triplet complementary to the codon on messenger RNA that specifies that particular amino acid. A ribosome assembles on the start codon AUG. Transfer RNA with the anticodon UAC and carrying the amino acid methionine binds to the codon. The transfer RNA is in the P site of the ribosome. The A site is available for a second transfer RNA with an anticodon complementary to the second messenger RNA triplet. The amino acid carried by the second transfer RNA binds to the methionine. The first transfer RNA leaves the P site, and the second transfer RNA moves there, still bound to messenger RNA. This brings the third messenger RNA codon to the now empty A site, and the appropriate transfer RNA can bind to it. The third amino acid is added to the chain, and translocation occurs once again. This process of polypeptide chain elongation continues until a stop codon is reached. A release factor binds to the A site. It carries no amino acids, but facilitates release of the polypeptide and the messenger RNA from the ribosome.
Now, another bonus.
Genetic code!
Genetic code. Degenerate. Having more than one base triple to code for one amino acid. Universal. Found in all living organisms. The relationship between one gene and one polypeptide. Polypeptides are long chains of amino acids. Amino acids must be linked up in a precise sequence to make a polypeptide. Genes store the information needed to make a polypeptide in a coded form. The sequence of bases in a gene codes for the sequence of amino acids in a polypeptide. The information in the gene is decoded during the making of the polypeptide. This process is conducted in 2 stages known as transcription and translation.
Okay, yesterday when everyone came back from Maya's party, guess what we did?
Biology!
We worked really hard for this video. We stayed up until 6 a.m. working on it.
Then we slept till 10, and then we
We
started
went
working
to
on
breakfast at my brother's girlfriend's house.
and then we came here.
We came to Sophia's house, and then we recorded these things, and we did the perfect video so you guys
Can learn
everything about replication, transcription, and translation. So you get the top grades in the IB exam. So you guys really have to thank us for everything.
You know what time it is right now? It's almost 7, and we've been working on this since
The whole day.
morning.
Yeah, so
We think
we
we
love
did a good
biology,
job.
and we think we deserve a good grade.
Thank you, we love you.
So clap, so clap, so clap, so clap, so clap, so clap, so clap. Yeah, whoa. Miss me want to see everybody on move. Dance, you know, that man coming at you. We just want to big up QTB. Bob Sinclair, it's a dance thing, you see me? Somewhere you just bounce up. Yeah. Everybody dance now. Let's go.
Hey, what's up? Testing 1, 2, 3. This is H Dog and Q Doggy Dog Dog DNA replication. Deox, ribonucleic acid is made from 2 strands on double helix. So then the strands come apart. Nitrogen bases they start to unzip by DNA helicase into protein, adenine, and thymine. H2 bonds between cytosine and guanine. H2 bonds, ladder structure is strong as in phosphodiestylate DN. A polymerase builds a new strand from now. 5 prime to 3 prime direction. H2 bonds between adenine and thymine. H2 bonds between cytosine and guanine. H2 bonds, ladder structure is strong.
My mom's down.